Student, today I am going to discuss two topics. One is Gpcr, which is popularly known as G-protein coupled reaction, and the role of the second messengers, that is cyclic AMP. So these chapters, these two topics belongs to the chapter cell signalings. Okay, so cell signaling means we know the cell. Every cell is having the cell membrane, and this cell membrane is impermeable or selectively permeable. It allows to pass some selective substances. Okay. Now, when it allows to pass some selective substances, there is some receptors are there, which is transmembrane proteins present in the cell membrane, which help sense what transactions of these different substances, as well as different messages from first messenger to the second messengers. Okay. So first, let us give regarding the introductions. Okay, the plasma membrane is impermeable to the water-soluble material, including ions, small inorganic molecules, and polypeptides or proteins. To enter or influence the cells, and hydrophilic material must react with the protein residents in the plasma membrane. The extracellular material is called the ligands. Extracellular material means hormone, enzymes, maybe drugs, maybe pathogens. Okay. Target protein to which the ligand bind is called the receptors. And receptors is always found in on the cell membrane. Such interactions are not necessary for lipid soluble molecules such as steroid hormone. In case of the steroid hormone, there is no requirement of this plasma membrane, because steroid hormone, by dissolving the lipid layer, it can enter and can pass to the cytoplasm, and then it will perform other activities. Okay. When a receptor remains in the membrane and responds to a ligand binding by triggering a response pathway in the cytosol, the process is called signal transductions, because a signal has an effect been transduced across the membrane. Signal transduction provides a means of amplification of the original signal. When an extracellular ligand interacts with the membrane receptor, it converts it from an inactive to an active form. In its active form, the receptor stimulates the catalytic activity that generates a cytosolic signal whose amplitude is much greater than the original extracellular signal. A molecule produced in response to the transductions is an extracellular signal is called the second messenger or cyclic AMP. By contrast with the first messenger, there is hormone, the ligand, which has the extracellular ligands. Okay. So in my next slides, just I would like to give the there is two major types of the signal transductions are reported. The first one is known the receptor may interact with the G protein that is associated with the membrane. G protein are named for the ability to bind the guanine nucleotides. Inactive from the G protein is a trimer bound to the GDP. Upon binding a ligand, a receptor acts upon the G protein to cause the GDP to be replaced with the GTP. As a result, the G protein dissociates into a single subunit carrying the GTP and the dimer. Either the monomer or the dimer that acts upon the protein, often also associated with the membrane, which in turn reacts with the target or targets in the cytoplasm. This chain of events often stimulates the production of the second messengers. The classic examples being the cyclic AMP. Another signal transduction is known the receptors may be transmembrane proteins with domains on both the extracellular sites and the cytoplasmic sites. Domains with element, that is cytoplasmic site as well as the that is extracellular sites. Okay. The ligand binding on the extracellular side. Extracellular means outside the cellular. Ligand binding on the extracellular side influences the activity of the domain on the cytoplasmic side. Once it binds, ligand binds to the extracellular side, automatically the cytoplasmic side is activated. Okay. A typical reaction is for ligand binding in the activity of the protein tyrosine kinase activity produced by a cytoplasmic domain. The kinase phosphorylates its own cytoplasmic domain. The phosphorylation enables the receptor to associate with the activity of the target protein, which in turn acts upon the new substrate within the cells. Now, receptors. Now, receptors on the cell surface. Okay. The cell surface receptors are following categories. Okay. One is in the ion channel. Ion channel means it is a protein resident, it is found, the channel is found, it is made up of protein, resident protein, it is found in the cell membrane. Okay. And may constitute the ion channel that control the passage of the ions. Ion means There's a sodium ion, potassium ion, chloride ions, bicarbonate ions, okay, that can pass through the ion channel, a very small molecule passively. It's ligand transport. Now, when ligand means is a large molecular weight molecules, maybe hormones, maybe enzymes, pathogen like that, ligand transport. Receptor mediated is the ligand mount. That ligands, when bind, the receptors are found in the again cell membrane, surface of the cell membrane. Okay. Receptor internalization. There is some ligand bindings may trigger the process of internalization in which the receptor and ligand combination is brought about in the cell by the process of the endocytosis and the exocytosis. So these are the three types of the receptors which are found in the cell surface. Okay. Now forms of the signaling molecules. Now signaling molecules also again of different forms. Okay. Sometimes they may be there is protein molecules. Sometimes they may be lipid molecules. Okay. Sometimes they may be amines. Okay. They may be of different categories. So cell to cell signaling involves transmission of the signal from a sending cell to a receiving cell. However, not all sending and receiving cells are next door neighbors, nor do all cell pairs exchange signals in the same way. There are four basic categories of the chemical signaling found in the multicellular organisms. Okay. First is the Parakan Signalings. Parakines. So let us discuss the Parakan Signalings. Okay. So Parakan means the cells that are near the cells that are near one another. Both the cells are nearer, okay, and communicate through the release of the chemical messengers. Okay, this type of the signaling in which the cells communicate over relatively short distances is known as the Parakan signaling. Parakan signaling allows cells to locally coordinate activities with their neighbors. Parakan signals are especially important during the development when they allow one group of the cells to tell a neighboring group of cells what cellular identity to take on. Okay. Another is known as your second one is known as the synaptic signalings. Okay. Now synaptic signaling, the synaptic signaling is a process of signaling in the junction between the two nerve cells. Okay, where signal transmission occurs. When the sending neuron fires an electrical impulse moves rapidly at the presynaptic node. When the impulse reaches to the synapse, okay, it triggers the release of the ligands called the neurotransmitter, for example, it is acetylcholine, which quickly cross the small gap between the nerve cells. The postsynaptic nerve at the receiving cells then bind to the chemical and causes the chemical changes inside the cells. So here is the diagrams, schematic diagram, by means of which you can understand what is a parakan and what is the signalings. Okay, Parakan signaling and is known as that is synapse signaling. Okay. Now my third diagram signaling that is known as the paracrine. So two cells are there. Okay. One is a bluish or greenish color and another is the reddish color. Now says signaling one cell it secretes some secretions red color. That secretion it affects the target cell. So this type of the signaling in a nearer cells, neighboring cells is known as the parakines. Okay, a cell targets the nearby cells, and that is in the Parakan signaling. And that's the second one is known as signaling at the gap junction, which is popular in the synapse. A cell targets a cell connection by a gap junction. Okay, so here what happens, two cells are connected with the gap junctions. Okay, it may be the gap junction or it may be synaptic connections. Okay, now this red one is known as some neurotransmitters. When it's transmitted, it will enter into the target cells. Okay, by means of gap junction or the synapse. So this is known as the synaptic transmissions. Okay, another that is known the autokine signaling. Autokine signaling means the autokine signaling a cell signals to itself, okay, releasing a ligand that binds to the receptors on its own surface. This may seem to like an odd thing for a cell to do, but autokine signaling plays an important role in many processes. For example, autokine signaling is important in the development, helping the cell take on and reinforce their correct identities. From a medical standpoint, autocrine signaling is important in cancer and is thought to play an important role in metastasis, the spread of the cancer from the original site to other parts of the body. In many cases, a signal may have both autocrine and paracrine effects, binding to the sending cell well as other similar cells in the area. So already I have shown the diagram. Just you can look the autocrine signaling in the earlier diagram. So autocrine means the first one. Same cell it secretes the say some secretion, some chemicals. That chemicals act on the same cell. So that is on the autocrines. Okay. Best example is the lysozymes. Okay. So autocrine. And next is after autocrine signaling and there is endocrine signalings. Okay. So endocrine signaling means already you must you come across endocrine means hormones. Okay. When a cell needs to transmit endocrine and then endocrines. Okay. When a cell needs to transmit signals over long distances, they often use the circulatory systems as a distribution network for messages they send. In long distance endocrine signaling, signals are produced by specialized cells and released into the bloodstreams, which carries them to target cells in distinct part of the body. Signals that are produced in one part of the body and travel through the circulation to reach the faraway targets on the hormones. In humans, endocrine glands that release the hormones include the thyroid, the hypothalamus, and the pituitary, as well as the gonads and the pancreas. Each endocrine gland releases one or more types of the hormones. Okay, many of which are master regulators of the development and the physiology. For example, the pituitary releases the growth hormones, which promote the growth and particularly the skeleton and the cartilage of the body. Okay, so hormones already endocrine glands secrete the hormones. Hormones are secreted by the one particular gland, and it will act in the target gland which is far away. Say pituitary gland it secretes the pituitary hormones, but pituitary hormones the somatotropic hormone, adrenocorticotropic hormone, then your thyroid stimulating hormones. Okay, all are pituitary hormones, but they will act. Thyroid stimulating hormone act on the thyroid gland. Okay, LH and FSH it will act on the your ovary as well as the your that is testes. People point. ACT is adrenocorticotropic hormone. It will adrenal glands, which is far away. Okay, so this type of the that is signaling is known as the endocrine signaling. Okay. In addition to that, there is another kind of signaling is there, which is known as the signaling through the cell-to-cell contact. Okay. Now already I have shown you gap junctions. Okay. Gap junctions in animals and plasmodisphoria in the plants are tiny channels directly connect neighboring cells. These water-filled channels allow some signaling molecules called the intercellular mediators to diffuse between the two cells. Small molecules such as calcium divalent ions are able to move between the cells, but large molecules like the proteins and DNA cannot pass through the channels without the special assistance. This allows a group of cells to coordinate their response to signal that only one of them may have received. And another from the direct signaling, the two cells may bind to one another because they carry complementary proteins on their surfaces. When the proteins bind to one another, this interaction changes the shape of one of the proteins, transmitting a signal. This kind of the signaling is especially important in the immunosystem, where immunocells use cell surface markers to recognize self cells, that is the body's own cells, and the cells infected with the pathogen of the foreign cells. Okay, so there is another kind of signalings. Now, once signaling is over, next portion is known the receptors. How this signaling or these chemical molecules or this ligand it will be accepted by the receptors. Okay, there are different kinds of receptors are there. So first, in my schematic diagrams, I am showing you that first on I have shown you ligand gated ion channels. That is ion channel signalings. Okay, receptors. Okay. Next, second one is known as G protein coupled reactors, which is given in your syllabus. Okay, there is known as GC, G protein GPCR. Okay, the G protein coupled reactions, and third one is the kinase linked receptors, nuclear receptors. Okay, now first one that is the ion channels. In case of the ion channels, what will happens? When any ions, it will pass through the that is channels which is present in the surface of the cell membranes. Okay, different points from the higher concentration, the lower concentration in a passive way. It will enter into the cell and it will perform the different cellular activities. So this is on the one kind of the receptors. Second kind of a receptor is the G protein coupled receptors. So this is a very complex ones. Okay, G protein coupled receptor. G protein is a one kind of the proteins which is a heptamer proteins which is found in the surface of the cell membrane. It has a one extra that is a cytoplasmic that is a domain as well as the cytosol domains. Okay, the two extracellular domains and the intercellular domain. Two types of domains are found, and it will help in the transport of the larger molecules, maybe hormones, maybe enzymes. Yeah. Okay, so the diagram it is shown here. Okay, so later we will discuss in details. So this is the G protein receptors, and this is the another kind of the receptors. Third one is the kinase link receptor. This is like a G protein, but it is not so complex. Okay, it is a simply one kind of dimers is there. Therefore, the dimer, the receptors are formed, and ultimately the molecules will be what it will be come inside the cytoplasms, and it will perform the different activities. Another kind of receptors is the nuclear receptors. Nuclear receptor actually it is applicable in the steroid hormone. Now steroid hormones are mainly made up of the lipid molecule, and lipid molecule it can pass through the cell membrane by simply dissolving the lipid layer of the cell membrane, and it enters the nuclear membrane also by dissolving the lipid layer of the nuclear membrane. Inside the nucleus there is some hormone receptor complexes are there which remain bind with the DNA. It binds with the hormone receptor complex. As a result of what will happen, it will trigger the DNA for gene transcriptions. Ultimately, one is triggered the DNA. DNA will give rise to the mRNA, and mRNA will give rise to the protein synthesis. This type of the effects that is known the nuclear receptors. Okay, so in my next slides, so I would like to give regarding the types of the receptors in details. Receptors which from the ion channel. An ion channel comprises the water-soluble pores in the membrane. The activity is controlled by the regulation of the opening and closing of the channel. When it is open, ion can diffuse passively as driven by the electrochemical gradient from higher electrochemical gradient to lower electrochemical gradients. Okay. Ion channels allow only passage of the passive transport. It is very passive transport. No energy, no carrier is required. The resting state of an ion channel is closed. The resting state of the ion channel is closed, and the gates that control the channel activity usually open only briefly in response to the specific signal. Ion channels are classified in according to the type of the signal that controls the gate. It may be gap junction, ligand gated, voltage gated, second messenger gated, etc. Okay. Now, this ion channel again it is ligand gated channels are receptors that respond to the binding of the particular molecules among with the neurotransmitter, the acetylcholine, glycines, gamma aminobutyric acid, glutamic acid are important. Voltage gated channels response to the electrical changes, again prominent features of the our neutral systems. Okay, and second messenger gated channel provides yet another means of signal transactions, receptors with the ion channels. So next I will discuss the G protein coupled reactions, which is the main importance, and it is given in your syllabus also, G protein coupled reactions. So what is the thing mean by the G protein coupled reactions? Okay, now try to understand very nicely. Okay, now G protein coupled receptors, which is popularly known as Gpcr. Okay, now G protein coupled receptor Gpcr is also known as 7 transmembrane domain receptors. 7 transmembrane domain receptor. That means in the cell membrane, this G protein it has got 7 helixes. Okay, that 7 helixes it crosses the cell membranes. Okay, so these are the 7 transmembrane domain receptors. It is also the heptahelical receptor because since it has the 7 that means helical structures, that is a popularity is also on the heptahelical receptors. It is also on the serpentine, just like receptor, that is the serpentine receptors and G protein link receptors. Okay, it constitutes the large protein family of the receptor that detect molecules outside the cell and activate the internal signal transaction pathway and ultimately cellular responses. Coupling with the G proteins, they are called 7 transmembrane receptors because they pass through the cell membrane in 7 times. Okay. The ligands that bind and activate these receptors include the higher, that is light sensitive compounds, odors, pheromones, hormones, and neurotransmitters, and vary in size and from small molecules to peptide and large molecule protein. Each G protein coupled receptors are consist of an extracellular, outside the cell, NS2 terminus or N terminus, 7 membrane spanning helices in the cell membrane itself, and the subatomic carboxy terminus COH. So there are two terminus. One is this N terminus, which found in the outside, okay, that is extracellular, and there is the C terminus, which is found inside, that is cytoplasmic terminus. Okay, the N terminus is glycosylated. Okay, the cytoplasmic loops of the receptors play a very important role in the hormone that is induced activation of the G protein. When hormone ligand molecule is attached to its receptor, one or more the cytoplasmic loops interact with the G protein and activate them. Now we see this is a very nice diagram regarding the G proteins. So each green one, there is a numbering, there is 1, 2, 3, 4, 5, 6, 7. It will be in the 7. There is a helix. Okay. And the first one, that is it has the receptor outside the cytoplasm. And last 7 one, it has got NS2. That is a COH terminal where there is alpha, beta, and gamma. Three there's a factor it remain binds with the G proteins. Okay. Now the first one, it is the inactive states. Okay. And there is a 1, 2, 3, 3 loops in the extracellular and 3 loops in the intercellular remain found. Okay. So this is the cell membrane, and these are the different, and there is the helixes of the G proteins. Now this is the inactive states. Okay. In the inactive states always there is along with the alpha, the GDP remain bounds. When some hormones, the red one is the hormones in the second diagram, hormones binds some hormones or some ligand. If you may say hormone, you may say ligand. Okay. When some ligand or hormones or some molecules is bind, okay, in the first that is the receptors. So receptor, what will happen? It will transduce some changes in the what? That is the interior of the cells. That is alpha, beta, and gamma. Beta and gamma will be separated. Okay, and alpha will be what? GTP will bind to the alpha. Okay, so this is the most important thing regarding the G protein couple reaction. The diagram just schematically I am giving. So in the next diagrams, just I am giving the details of that is the G protein transaction signals from the variety of the receptors or the variety of the targets. The components of the general pathway can be described as a receptor is a resident membrane. Resident membrane means inside the membrane it is found and is a resident. That protein is a resident protein, is not the outside protein. A receptor is a resident membrane protein that is activated by an extracellular signals. Extracellular signal means hormones, enzymes like that. A protein is converted into a active form when the interaction with the activated receptor causes is bound to GDP to be replaced by the GDP. When it binds in the there is a NS2 terminals in the C terminal, there will be changes. Okay, and effector is in the target protein that is activated by the G protein. Sometimes it is another membrane associated proteins. Okay, it will effector is just a adenylyl cyclase systems. So ultimately what happens? It will affect it will trigger the adenylyl cyclase systems. Okay, active, that is the alpha unit along with the GTP. A second messenger, as a result, what will happen? A second messenger is a small molecule that is released as a result of the activation of the certain effectors. So there is a 4 part in case of the transduction of the G proteins. Okay, that just signals from the outside to the inside. So the 4 there is a part are there. Now another terminology used to describe the relationship of the components of the receptor generally known the upstream of the G protein, while the effectors are known the downstream of the G proteins. Now G protein are trimers, that is inside trimers, whose function depends on the ability to dissociate into alpha monomer and beta and gamma dimer. Alpha subunit binds the guanine nucleotide and the reaction control association with the beta gamma dimer. And alpha subunit bound to the GDP associates stably with the beta and gamma to form the inact the G protein. In the trimeric state, G protein is active. Now inside the G protein, what will happen in the cytosolic part? The 3 units are there, alpha, beta, and gamma. When 3 units remain bind with the G protein, then it is the inactive states. It is not working. Okay. And during that stage, again, alpha unit is that is associated with the GDP molecule. Now, as and when some ligands or the first passenger binds to the extracellular receptor or the G protein, GDP and alpha subunit dissociate from the trimers. Okay. GDP and alpha subunit dissociate from the trimers. GDP converts into GDP with the alpha subunit, which now binds to the effectors and activate the adenylyl cyclase system in the cytosol. Displacement of the GDP by GTP generates this alpha GDP subunit, which dissociate from the beta gamma dimer may attach to the any G proteins. Okay. Now that's beta gamma bit may attach to the another G proteins. Okay. Now adenylyl cyclase systems is what it is the another part of the protein, another protein molecule which is found in our that is a cell membrane. Now converts ATP to cyclic AMP. Okay, it converts ATP to cyclic AMP or the second messengers in the cytosol. Now cyclic AMP activates inactivates that is activates inactive protein kinase into the active protein kinases. Okay, as a result, it triggers responses of the target cell, activates the enzyme, stimulates cellular secretion, open ion channel, etc. Hormones that act via the cyclic AMP mechanism are epinephrine, glucagon, ACTase, PTase, FSH, LS, TSH, and calcitonins. Okay, so this is the main mechanism how the G protein ubiquitin acts. Now, in this schematic diagram, it is also given. There are 5 steps. It has been shown in these schematic diagrams. Okay, now you see that is the first the diagrams. Hormones of the first messenger binds to a G protein. It is outside the cell membrane. Lipid bilayer, this is the G protein. So when the hormones that is the first messenger binds to a receptors, the G protein. Okay, so there is a that means a trimer. The trimer is not shown here in the G protein. Out of the 3 trimer, the one trimer will be activated. That is on the alpha unit, stimulating. It will be stimulated. When it's stimulated, GTP will bind and it will be separated from the original G proteins. Okay. Okay. Now receptor activate the G protein. That is once it activate the G protein, it will be separated from the main G protein, and you will trigger G protein trigger or it stimulate what the another protein which is known as adenylate cyclase. G protein activate the adenylate cyclase. Now, adenylate cyclase is also found in the cell membrane. Now, when it is activated with the help of this binding of this GTP and alpha unit, what will happen? Then it will convert inactive enzyme, that is cyclic it will be liberate, that is activate, and that is adenylate cyclase system. As a result, it will form a cyclic AMP. Okay, converting the ATP on the cyclic AMP. That's the second messenger. Now cyclic AMP, what will happen? This second messenger now will activate the different protein kinases, inactive protein kinases into active protein kinases. Okay, triggers the response to the triggers, target cell, activate enzymes, stimulate the cellular activities, open ion channel, etc. So in this way, what happens? G protein coupled reactions takes place. Okay, so this is the schematic diagram. Okay, here all the units are not shown, alpha, beta, and gamma. It is very difficult. That is not shown. Only one unit it has been shown. That is the alpha unit. Okay, the beta gamma, what will happen? It will be separated and it will bind to the another G protein. So that again another G protein will remain in the there is inactive states. Okay, now with the help of this second messenger, what happens? Hormones that act this is a cyclic mechanism. Epinephrine, glucagon, ACTase, PTase, FSH, TSH, LHS, and calcitonins. All these hormones are what it will be transmitted, and it will what happens? It will perform its activities. Okay. So in my next that is slides, just I would like to show another thing, that is G proteins. That is known as the G protein alpha subunit together with the bound with the GTP can then dissociate from the beta and the gamma subunit to further affect the intercellular signaling protein or target functional protein, that is directly depending on the subunit type. Okay, you follow my point? So now there are 2 principal signal transaction pathway involving the G protein coupled reaction. One is cyclic AMP signal pathway, and that is phospho inositol signal pathway. Adenylate cyclase already have shown activity is activated when the binds the sub into the activated G protein. Activation of the cyclase ends when the G protein returns to the GDP state. As a result, there is a formation of cyclic AMP of the second messenger, which remain busy with the cellular activities. Is cyclic AMP triggers a chain of reaction which mainly include the modification of the one enzyme by another, which is known as cascade reaction. Now, adenylate cyclase may be activated or inhibited in another way, that is, for example, calcium or calmodulin binding, which can modify the activity of these enzymes in an active or synergistic fashion along with the G proteins. Now you see Gpcr, how Gpcr is activated with the help of the 3 timide is shown. The G protein, Gpcr, it remain bind. Okay, it remain bind with the G alpha, beta, and gamma unit. Okay, when there is some this ligand will bind, then this G alpha, it will be what separated from the gamma and the delta, and gamma delta it will attach to another G protein, and this alpha unit it will what activate the inner cyclic systems. Okay, the effector systems. So in my next diagrams. So this is another interesting diagrams regarding the different mechanism of the G protein. So there is a different stages are there. So first is the resting states. First stage the resting states. Then when what happened, alpha, beta, gamma is there outside and inside is there. Very nicely this shown. So when a ligand binds, what happens? This alpha, beta, and the gamma. Alpha unit and beta gamma unit will be separated. Okay. Now beta gamma it may bind to the another effectors. Okay. And it may help in the signaling, and this alpha unit bind to the another that is effector. It may also stimulate the signalings. Okay, so one big must that is binding is signaling is over. That signaling is known as formation of the there is a cyclic MP. Okay, when activation of the GTP is what will happen when again it is separated from these effector regions. Okay, GTP hydrolysis will take place. Then that is ligand bound is again separated. This okay. Then it will come to the original state, then active states with the binding with the 3 timers. Okay, so this is the simply schematic diagram. I'm showing all these things. So this is a very simple diagram regarding how the ligand bind, and this ligand helps in the cyclic system. And the cyclic system it converts it into cyclic MP, and cyclic MP activate the that is inactive to and protein kinase inactive to the active, and I perform the cellular activities. Okay, so same diagram it is through the ligand bindings. There is another 2-3 tag, and there is a PIP2. It is not in the syllabus. This is one kind of that is IP3 and your PIP2. Also, what there is one kind of the that is second messenger which also perform different activities inside the cells. Okay. Now, protein or polypeptide hormones already have ligands, maybe protein or polypeptide ligand, maybe steroid. Okay. Now, if the ligand is protein or polypeptide, then it will always what it will be carried or it will be transported by means of binding with the receptors. Okay, binding the receptor, then transduction, then system activations. So this is, and then there is the formation of the second messengers. Okay, second perform different activities. So this is in case the proteins, but sometimes only there is some steroid hormones also there. Not always proteins are there. Steroid hormones there. If steroid hormones receptors are proteins, okay, and that have a binding site for a particular steroid molecules. Okay, the steroid hormone receptors are bind in the cytosol and also in the nucleus of the steroid hormone. It can dissolve the cell membrane because it is made of the lipids. The cell membrane is also made of lipid layer. Directly by dissolving the lipid bilayer, it can enter the cytoplasm and binds in the nucleus of a target cells. Okay, steroid hormone receptors belong to the nuclear receptor family. They are ligand activated proteins and that regulate the transcription and selective genes. Their response elements are DNA sequences that are bound by the hormone receptor complex. The meaning is that steroid hormone directly it can cross the cell membrane. Okay, it enters in the cytoplasm. In the cytoplasm, there is some maybe receptors. Okay, if there is no receptor, then it can cross the your nuclear membrane. After crossing the nuclear membranes, there is a receptor is found on the DNA, which is on the hormone receptor complex. Okay, directly bound to the hormone receptor complex. Once it binds to the hormone receptor complex, it will trigger the or stimulate the DNA for transcriptions. So DNA ultimately gives us the formation of the mRNA, and mRNA ultimately gives us the protein, and protein it will perform the different activities. So this is the steroid hormone receptors. Okay. Next, this is the diagram how the steroid hormone receptors are working. Just I am showing. Directly it can cross the cell membrane, or it may with the steroid hormone it may bind to the hormone receptor complex, be in the cytoplasm, or it may be in the nucleus. Okay, and ultimately it will trigger the your DNA, activate the DNA, and it results in the formation of the transcription. And transcription ultimately again there is a protein synthesis will start. This is the steroid hormone how they act. Now, in addition to that, G protein, there is another type of the protein is the tyrosinase kinase receptors are there. Okay, so though it is not your syllabus, but this is a very important thing. There is a given in manuals. Okay, many hormones, for example, insulin-like growth hormone factors, epidermal growth factors, platelet-derived growth factors, fibroblast growth factors, colony-stimulating factors, etc. act on the target cell by binding the membrane-bound receptor which possesses tyrosine kinase activity. The receptors have an extracellular N-terminal, a single transmembrane helix, and a C carboxyl domain. The N-terminals contains a site of the glycosylation and the hormone binding site. The glycosylated site are not essential for the hormone binding. Okay, however, they are probably required for the proper processing of the receptor. The N-terminus has also been found in the rich in cytosine residues, which might be helpful in the aggregation as well as the in recycling of the receptors. The transmembrane domain seems to play a passive role in the signal transduction. However, it might be involved in the ligand induced internalization of the receptor. The C domain, the carboxyl domain, contains the several phosphorylation sites and tyrosine kinase activity. So this is the diagram of the yoke that is regarding the how tyrosine that is kinase activity by there is receptors, some receptors in the tyrosine kinase activity receptors, how they act. Now here two receptors it will be shown. So it is a always remain in the dimer states. Okay, so it may bind to a particular ligands. Okay, and bind means a rash or there is a MAP kinase signal pathway, it will activate. Okay, and there is a cytokines. If it binds, then with the JAK or STAT, it will activate JAK-STAT signal pathway. It will act. Since it is not in your syllabus, I am not giving in details. I am not discussing details. Okay. Simply you try to remember that in addition to the G protein coupled reaction, there is another type of receptors are there, which is popular known as tyrosine kinase. Now this is the diagram of the details of the tyrosine kinase, how they perform the activities. Okay. So these diamonds bound with the ligands. Then these diamonds receptor chain. Then what will happen? It will activation of the tyrosine kinase domain, and ultimately downstream action will start. Okay. This is the dimer. This is the monomer. Whatever it may be. So this is the idea how the tyrosines, that means the kinase enzyme, it performs different types of the activities. Okay. So this is the same thing regarding the diagram of the tyrosine kinase activity. Now receptors, now with the tyrosine kinase activity, can be placed under the three groups. Receptors, the epidermal growth factors, which have three domains: an extracellular N-terminal domain, a transmembrane domain, and a carboxyl terminal cytoplasmic domain. Receptors of the insulin and insulin-like growth factor 1, which have two domains. The extracellular N-terminal system, the alpha subunit, and the cytoplasmic domain is called the beta subunit. The alpha and the beta subunit linked together with the disulfide bonds. This is the insulin-like factors. And if the receptors are platelet-derived growth factors, okay, and colony stimulation factors, the binding with the ligand to the extracellular domain generally induce the dimerization of the receptors with the tyrosine kinase activity. The process of dimerization of the receptors leads to the trans-phosphorylation followed by the autophosphorylation of the cytoplasmic domains, resulting in increased tyrosine kinase activity of the receptors. The increased activity of the tyrosine kinase due to the phosphorylations catalyzes the phosphorylations and activation of specific Synaptic Signaling protein. These phosphorylated active proteins act as an important components of the hormone-induced signal transduction pathway, which finally culminate in the biological effects. With that, I would like to conclude. Okay. Do you follow my points? Now, if there is any problems, let me know. You are not responding properly, so try to know. Okay. With that, I would like to conclude.